Okay. It is my pleasure to introduce Professor Rufin Vogels as our first speaker at the symposium. Professor Vogels basically got his master's and PhD degrees in University of Leuven in Belgium in psychology. He's a full professor in the same institute currently. He has been recipient of many awards in Europe. He's the editor of Journal of Neuroscience and multiple other journals, including Scientific Reports. And he is one of the leading experts in basically ventral stream visual system with a variety of techniques that he's used in the past, including electrophysiology and fMRI, and especially the work with macaque monkeys. You know, from V1 to anterior IT, and face patches and body patches. And today's talk, the title of the talk, as you can see, is Body Representation in Macaque Inferior Temporal Cortex. With that, I would like to ask Rufin to start his presentation, and hopefully we can all learn from him at any time. Thank you.
Thank you for this nice introduction. So I'm going to review today some work that we did the past 10 years on body representations in the macaque inferior temporal cortex, which basically we use as a model to learn more about how the human brain processes visible images of bodies. As all of you might know, there's lots of research on neural processing of faces. We know that there are face category selective regions in the human brain, and the fusiform face area, occipital face area, and so on. Also in monkeys we have these so-called face patches, and there's also EEG work, ERP work, and so on, lots of work on face processing, but little work basically on body processing. Although neural processing of images of body is also pretty important. It's pretty essential for social behavior and for survival. At the individual level, also as a species, because non-verbal communication is partially based on analysis of body features, not only on face features, but also on body features, as you can see here, for instance. So we recognize to some extent what these people want to communicate or express. That not only holds for humans, but it also holds for our favorite species, which is macaque monkeys, rhesus monkeys, and they also signal through their posture their emotion, affects, and so on, and they express several types of displays, which are very important socially for their social behavior. And that's based on posture, it's based on their body posture. We also know from previous work that the coding of body posture also contributes to recognition of actions. So even from static images, you can do a posture analysis and recognize some action that might take place. But when you have a movie of the displays, so when there is dynamic body display, then even more information is presented with respect to actions, and so they are involved. There is a lot of action recognition based on posture information. So that's why it's also important to understand how bodies are processed by our system, by our visual system, and not only faces. Now we know from human fMRI studies and also from monkey fMRI studies that there are some regions especially in the temporal part of the brain, cortex, which are activated more strongly by images of bodies compared to faces. You have one here in occipital temporal part, which is called the EBA. It stands for extra striate body area, and then there's one more anterior here, which is the FBA, which is in the fusiform gyrus, so it's fusiform body area. It just a little bit posterior, partially overlaps even FFA, which is the face, fusiform face area. And the EBA is relatively close to the OFA, which is also a face area. So that's in humans. Now we know from some earlier studies in monkeys who used fMRI to look at category selective activity in their brain, that are also so-called body patches in rhesus monkeys. And already several years now, we started with a study in which we wanted to map these body patches using a variety of images. So we had images of human bodies. These are basically more or less the same that were used in the original human fMRI studies. And note that these are bodies without a head. I'm going to return to that issue later. So they're a little bit unnatural because they don't have a head. But we do that, and also in other studies that has been done, because we want to avoid phase activations. Then we have a set of monkey bodies, also without head. Sometimes they look a little bit weird, like this one here. Then we had four-legged mammals and birds. So these are all images of bodies. Then we had control categories. We had a set of human faces, monkey faces, different viewpoints, different expressions also. The same holds for bodies, basically. Then we had a set of sculptures. They were inspired by sculptures from Moore, an English artist. They're a little bit body-like, although, yeah. And then we had a set of fruits, vegetables. Then we had two sets of objects, control objects. And why do we have two sets? Well, the human bodies and the monkey bodies, they differ in low-level features, for instance, in their aspect ratio, which is plotted here. So this is the mean aspect ratio. And you can see for the human bodies, much larger than for the monkey body category. So so it was difficult to do this, to have one control category of objects for the human bodies and the monkey bodies. So we designed two, one which was matched in aspect ratio and also other low-level features with the monkey bodies, and the other with the human object category with the human bodies. So then we scanned these monkeys in using block design. And basically what we saw, and this is a flat map of the cortex of the monkey, or the posterior part. This here is the superior-temporal sulcus. It's also where many of these face patches are located, all of them, many of them. So this is V1 here. So this is superior-temporal sulcus. And in the lower bank, you'll see this here in blue and in red. These are data of two different animals. Plotted on top of each other on a standard brain. You'll see one body patch in the mid-STS, close to two face patches, in fact. But we'll talk about that more later. And then there's one more anterior, which we call then the anterior STS body patch. So we have the mid-STS body patch, which we call MSB, and then we have more anterior ASB. There is also one which is not shown here, and we don't see it all the time. There's also one here which is not in the STS, but a lot more ventral and medial, also close to another face patch. There is even more the third body patch. And sometimes we see also one little bit more posterior, close to an area called FST. Okay, now here in monkey, we have these body patches, and so the nice thing about the monkey is that you can do then single unit recordings to find out what these neurons are doing in these body patches, what kind of features that they respond to. So we recorded single unit activity and also local field potentials using the same electrode, basically. So from MSB initially, and then later on for ASB, and then we compared the two regions. And I'm now going to present some local field potential data of the of both body patches. So the MSB, the posterior one, and the more anterior one, and then some single unit data. Okay, so let's first look at local field potentials. local field potentials, which measure basically potentials close to where your electrode is. They're relatively local. They're not that local, but they're relatively local. And what we did to analyze these local field potentials is we computed the power as a function of frequency, for different frequencies, and a function of time. So the stimuli were shown for a relatively short duration, only 200 milliseconds. This first dotted line is stimulus onset, the second is stimulus offset. So this is the period during which the stimuli were shown. And so we had monkey faces, monkey bodies, human bodies, and so forth, all these different categories. These are data of one animal, this from a second monkey. Now if you look to the high frequencies here, so in the gamma range, above 50 Hertz, and that relates pretty well to basically multi-unit activity, to actual potential spiking activity. What you see is that you have, on average, stronger activation for the bodies, the monkey bodies, the human bodies, the mammals, and the birds, compared to the other categories. Also here in this second monkey, it's pretty strong, in fact. So you see that there seems to be, at least at that LFP level, at the power in the gamma range, you see a nice category selectivity which fits the fMRI data, in the sense you have a stronger activation for bodies compared to other object categories. So what about spiking activity? What about single units? Let's look now at spiking activity, action potentials. This is ASB and ASB, you find the same thing. So anterior you have the same thing. Also strong activation for bodies compared to non-bodies. But let's look now at spiking activity. This is this posterior or mid-STS body patch. This is a population peristimulus time histograms. So this is onset of the stimulus, this is the offset of the stimulus, and this is normalized firing rate. So for each stimulus, basically, we divide the response by the maximum response of the neuron to the set of stimuli. And then we average across the different categories. And so the blue to yellowish one here, blue to green yellow, these are the object categories. And you see on average there's a higher response than to the non-bodies. The sculptures are somewhat in the middle. This is also in the second monkey, especially here a little bit later. So also if you look at the average activity across the different images of these categories, we see a stronger response for the body categories compared to the other categories, which fits the gamma of the LFP and also fits the fMRI. So we see we have stronger, on average we have stronger activity for bodies compared to non-bodies. This also holds for the anterior body patch, for that ASB. It's a different color here. It's more the reddish one, or the body categories, and you see it's higher. So when we average now all the body categories and compare it to the non-body categories, which is here in dashed lines, and the full lines are the body. And so the average across the images of the 4 body categories, then we see in both regions, MSB in red and ASB in blue, we see stronger activation for the bodies compared to the non-bodies. What was interesting and somewhat surprising for us, in fact, is that if you look at the onset latency of the response, there is not much difference between, in fact, there is no real difference between MSB and ASB. There might be a tendency, small tendency to have a little bit earlier response for MSB compared to ASB, which was somewhat surprising because ASB is more anterior than MSB, and so hierarchically, we expect it to be at a later stage. So we expect that ASB gets input from MSB, but we didn't see clear difference in latency. However, when we look at the selectivity for bodies compared to non-bodies, then we see some differences. So that the selectivity, the body selectivity, is somewhat earlier in MSB compared to ASB. Now also note that also for non-body images, you can have a response. It's not that there is silence. I mean that there is above baseline activity. This here is baseline, above baseline activity for non-body images. These body patch cells don't respond exclusively to images of bodies. Okay, now these are all averages. averages across the different body images, the different object face images, and so on. But what happens then, and averages across neurons, all the different neurons, but what happens now at the individual single neuron level? Are all neurons really body selective? Is it the case that all neurons respond equally well or very strongly to all bodies of a category? Well, this is basically how it looks when we look at all individual units. What does this mean here, or what do we represent here? So each line corresponds to the activity of a single unit. Okay, so like in this case we have like 130 single units. So each horizontal line corresponds to a single unit, and then each vertical line corresponds to a particular stimuli. So the first 10 here are faces, monkey faces, and we have human faces, and so on. So these here, these are all bodies. These are responses to bodies. And you see that on average, this is the average, there is a stronger response to the bodies, to the body images, even individual body images compared to other objects, but all other non-body images. But also, you have some non-body images to which you also have a pretty good response. For instance, to this one, you also can have sometimes a pretty good response. Also, we have some faces to which we sometimes have a good response, especially this face here. And this is interesting because this is a face, but it has an interesting hairstyle in the sense that if you look at the hair of that lady, it's like two limbs basically, two extremities, like you also can find in the body, right? And in fact, when we remove the face, we did that for a couple of cells, we removed the internal features of the face, so we just kept the hair basically, then the neurons were still responding pretty well. So you can have response to faces, but probably not to the same kind of features as for face cells. But what is also striking, and that's in both monkeys, is that there is large heterogeneity. So some of these cells respond particularly well to some body images, but doesn't respond at all to other body images. And that varies from cell to cell, which kind of body image that they prefer. Note that body images, well, they're pretty heterogeneous, more heterogeneous than faces. And then images of faces, because they can differ many types of features, you have different viewpoints, different postures, and so on. So I have a large heterogeneity. That probably explains also heterogeneity of the single units. So basically, when we talk about body selectivity, we're talking about the average. We're talking about the average, the response to the average body compared to the average object or face. Because if you look at individual images, you can have a response to a body or not a response to a body, and even you can have a response to non-body images, like for instance to this one. Okay, so it's an average property. Basically, one could say that body selectivity in that body patch reflects a bias towards a higher response to body images compared to non-body images. These cells are for sure not semantic body detectors. Okay, so in ASB, so anterior one, we had the same picture. So also on average, you have a strong response to bodies, and also to some non-bodies here. On average, stronger to bodies compared to non-bodies, but there is still some pretty heterogeneity, as you can see. Now, an important question was, well, what are the kind of stimulus features that these cells respond to? Based on the previous results, or all the results I showed you so far, one could infer that there must be features that happen to be a lot or happen to be common in images of bodies and are less common in images of non-bodies, like in faces or everyday objects. So one of the first questions we addressed is, well, how important is shape, overall shape, in the sense of, well, would you have a similar response to a silhouette compared to the original image in which you have shading information, you have fur and well, texture and so on. Well, let's say we removed the texture, we removed all the internal features, we just keep the outline by showing the silhouette of the shape. Now, and somewhat, well, surprisingly, although as I'll show in the next slide, it might, excuse me, not be that surprising, is that overall the selectivity for the silhouette shapes, for the silhouette stimuli, was very similar to that for the original images. And that's shown here. This is average across a large population of neurons. And for each neuron, we ranked 10 images from its best to its worst. So for each neuron, then we averaged that across neurons. So you see that strong selectivity you have. For some images, you have a nice response, for others not. And then we applied the same ranking for the silhouette. And you see, you also have there a nice decrease in response, showing that the overall shape preference is preserved when you go from original to silhouette shape. So these MSB neurons, they respond predominantly to shape features. I'm not claiming here that they only respond to shape features, because there are some differences if you look at the individual image level, in differences in response. But overall, shape features are pretty important. We haven't done this test for anterior body patch, but Doris, in laboratory of Doris Sauer at Caltech, they have done a similar analysis, in fact, at the fMRI level, and they also showed that even also in the anterior body patch, you have strong activations to silhouettes, which fits basically the original images. So also it seems to hold true for the anterior body patch. Now, at first we found it surprising, but well, maybe it's not that surprising, because as you can see here, shape features and silhouettes, they're largely sufficient to detect and discriminate body, unlike body posture, as you can see here. Okay, you can recognize that this is a monkey. This here is even a different species of this monkey here, and so you can recognize different postures just by the silhouette. Again, it doesn't mean that other kinds of features, for instance, shading and texture, and also features that signal depth, like binocular disparity, they don't contribute to the response or selectivity in these body patches. But predominantly, I think shape is an important factor. Okay, so shape being important, then of course it's still interesting to know what kind of features, because you have different kind of shape features, are determining that response. Frankly, we still don't know. I cannot give you the full answer. We did a couple of experiments addressing that, and which gives a partial answer. And in one of the experiments, which I'm going to review briefly here, we used so-called Vogels technique, which was developed by Philip Shins many years ago in the context of psychophysics, human psychophysical experiments, and also then later was employed in analyzing EEG and MEG data. And we also employed it here in collaboration with Philip Shins to try to get a handle on what kind of image features are determining the responses in these body patch neurons. And I'm talking here only about MSB, so the posterior, the mid-STS body patch. So we start from an image to which the neuron responds very well. Right? Let's say it's this image of this lady here without head. And then what we do is we do a spatial frequency decomposition. So we filter the image in different spatial frequency bands. So here high spatial frequencies to very low spatial frequencies. So this is very blurred body here. Here you only see the, well, let's say the contours and the edges of the body. And then we sampled these spatial frequency filtered images. We sampled them by so-called bubbles. And these are like Gaussian apertures. So these are Gaussian apertures through which you see the original image. So basically we partially occlude this image. We only can see it through some small apertures, roundish Gaussian apertures. And we have many of them for the low frequencies, and we have less of them for the high frequencies, and less for them for the low frequencies. And then we we apply the bubbles, these apertures, and then we add all the frequencies together and get an image like this, in which some parts of the body and some frequencies are remaining and others are gone. And we generate like that thousands of images, okay, and those we present to the cell. And then we use so-called reverse correlation, so technique, in order to see which part of the image needs to be present in order to get a good response from the neuron. I'm not going to go here into the details. You can read it in the paper and also the publication that used that Vogels technique. So we have many of these stimuli in which always different parts at random are present at different spatial frequencies. And then when we have a good response of the neuron, we can see what image was presented. And so we add up all the images basically that provided a good response. And by doing that, we can figure out, can get a handle to which features were driving the response. And that we did for the original images and also for some silhouettes. And these are some of the data we obtained. So colored here in orange and red are the features that were activating, were driving, according to the Vogels technique, were driving the response to the neurons. For instance, here it was a part of a leg, trunk of an elephant. Interestingly, this was a cell that responds pretty well to this object here, which is obviously not a body, but it has some limbs, it has some extremities. So the cell was responding to that. Some cells were responding to curved boundary segments, which are parts of the bodies, but sometimes also part of a non-body, as you can see here, and so on. Few of the cells required the full body in order to get a response. So most of these cells, the large majority of the cells, were happy with only parts in that MSB. And most of them preferred extremities. Now we followed this up, and that idea that not a full body is necessary to drive the response in MSB, but only parts, by presenting body parts alone, like here, and compare that to the full body. And then we saw that for each cell, basically, we could find the best, a very good body part to which the response was more or less equal to the full body. So what's plotted here is an index, which is response to the part minus the response to the full body divided by the sum of the two. So this index will be 0 when you have an equal response to the part and the full body. And you see for some parts, which were basic, and I must say they were determined in an unbiased way, using half of the trials, not to get the circularity here. These are technical details, is that this index here is close to 0 for the best body part. So you can find the best a body part, only a part, usually what that was an extremity like a limb, to which a neural response as good as to the full body. And there's also some other part to which you nearly don't have any response. So MSB responds well and selectively to body parts. We have some other evidence that the same holds also for ASB, but we didn't do a strict comparison between MSB and more anterior region ASB. Okay, so another, and that was not unexpected, of course, is that it doesn't matter where that feature is located within the visual field. The cells have receptive fields, so it matters to some extent, but you can translate it. You can even translate it to the object or the feature to a completely non-overlapping position in order to get a very good response. So if you present your images at this location or you present it at that location while the monkey is fixating here, you get basically the same feature. You have pretty high correlations between these maps and these maps. Same holds for size, so there's also size tolerance. So you can decrease the size by 50% and still get pretty good overlap in the features that you obtain using the bubbles. And also if you do other experiments in which you don't use bubbles but just vary the size or vary the position, you see the classical feature of IT, basically that these cells show strong position and size tolerance. These body cells, they are not they're not an exception to that rule. Now, to get now some handle on the differences between MSB and ASB, because we have these two patches to see whether they are doing the same, basically, because so far we didn't see much difference in in all the data I showed. Thus far, it was pretty similar. But in order to get some handle, we created a new stimulus set of monkey bodies, monkey avatars, basically. And they consisted of 3 identities, 5 poachers, and we ran it on a different viewpoints. And we basically looked at how these cells encoded identity, poacher, and viewpoint. So these are the avatars that we used. And they have a face, but the face everywhere, the face is the same basically. So it's the same face identity, and it's not very well visible, as you can see, the facial features. So it's mainly the body that is supposed to drive the response. So we had 3 identities. We had a fat monkey, a normal sized monkey, and then a thin monkey. So we used basically the body mass index as identity. And then each identity was run at these different viewpoints, and each identity could have 5 different postures. And these were natural postures based on an ethogram that was developed, was made based on ethological observations of monkey behavior by a ethologist many years ago. So these are really, ethologically relevant postures that we used. So we presented these stimuli, so we have 120 stimuli, 8 postures times 5 postures times 3 identities to our cells. And this is a response of an MSB cell, so the mid-STS, the posterior face patch. So each column corresponds to a viewpoint, and each row corresponds to a posture. And this is here for one identity, for the normal identity. And you can see you have a strong selectivity for viewpoint. So these cells are strongly selective for viewpoint, and also show some selectivity, pretty good selectivity even for posture, and also for identity. So basically they have they're pretty selective for basic image properties, let's say. If you then go to ASB, and this is a typical ASB neurons, they are also very selective for posture, but they are less selective. They still show some selectivity, but less selective for viewpoint. They generalize, for instance, across these mirror views here. So there is less tolerance for viewpoint, and that turned out to be a general characteristic. In order to show that general characteristic, I'm going to show you some results in which we decoded the identity, the posture, and the viewpoint from the responses of these neurons. I'm not going to go into the technical details, so we used the classifier basically to classify the identity of a stimulus based on the response of a set of neurons on a particular trial, presentation, and the identity should be classified independent of its posture and viewpoint. And we did the same thing for the posture and did the same thing for the viewpoint. And we used linear for the people who were interested in technical details, well, it's all published. We used a linear support vector machine as classifier, and also 6-fold cross-validation. So the training and test sets, they were independent as a standard in this kind of analysis. Now, before going to the identity, posture, and viewpoint decoding, I'm going to show that there was no difference in MSB and ASB when we asked the classifier to identify which image, which of these 120 images was presented. So the overall image selectivity is very similar for MSB and ASB. However, when we asked the classifier to decode identity, then MSB was worse than ASB. So the posterior patch is worse than the anterior patch. And this is identity irrespective of viewpoint, irrespective of posture. The same thing for posture. So when we ask the classifier to classify posture, irrespective of the identity, irrespective of the viewpoint, then ASB is better than MSB. However, when we ask the classifier to classify viewpoint, then the performance is better for MSB compared to ASB. So basically, what we see here is that the representation becomes or tolerates more differences in viewpoint as one moves anterior, and as a result of that, we also get better classification or better representation, encoding of identity and posture irrespective of the viewpoint. Okay, so I'll make some interim conclusions here, and after that I'm going to present some work that we are doing currently. In fact, at this very moment, as I'm talking, we are scanning a monkey in one of these experiments. There's an fMRI experiment going on at this very moment here. And these are some preliminary data, but I want to share them with you anyway. Okay, but so some interim conclusion is, so we have these fMRI-defined patches in the superior temporal sulcus of the monkey, and the mid-STS, MSB, anterior STS body patch, ASB, and they, on average, they show a great response to bodies compared with other categories, including faces. But they have a strong within-body category selectivity. But that I mean that they don't respond to all images of bodies. They respond to some images and not to other images. For instance, in MSB, we saw that there is a strong dependency on the viewpoint and also on the posture, also in ASB. So they are not body detectors. They show selectivity, strong selectivity within the body category, which makes them very suitable for analyzing basically body shape. So these are not semantic body detectors, but they respond to features that are prevalent in images of bodies, and sometimes when these features are present in non-body images. I show some examples of those. You can also have a response to these non-body images. So these are not semantic body detectors. They respond to features that happen to be present a lot in bodies. And then I showed that there is a difference between these body patches, between MSB and ASB. You have a greater encoding of identity and posture that's tolerant to changes in viewpoint in ASB compared to MSB. And you have a greater sensitivity to 3D orientation MSB compared to ASB. And this is, in fact, very similar to what was shown by Winrich Freiwald and Doris Sauer in the face patch system. Also, when you go from the posterior faces to the anterior face patches, then you see that there is an increase in the tolerance to differences in face viewpoint, and you also have better decoding of face identity, more anterior than posterior face identity, irrespective of the viewpoint. And in fact, there is very recent work published last year showing that also for some other object categories that might be the same, that might be a general principle within the inferior temporal cortex, that you have a more viewpoint invariant representation or tolerant presentation to more anterior to the system. Okay, but as I told you before showing this conclusion slide, I would like now to share you some very recent experiments or data that we are running right now, basically. And the first one has to do with the the relationship between these faces and body patches. And what's shown here, these are data of one monkey in which we show the face patches, defined here by faces minus a set of objects, and the body patches, bodies minus a set of objects. These are in fact different images than the one I showed you before. We created a new stimulus set, but that's not that important. The results are pretty similar. And so in red, so you see face activity, face-related activity. In blue, you see body-related activity. And you see there is some partial overlap, but there is also distinct activation. Also note that there is that the face and the body patches they overlap. Overlap. Sorry, they're neighbors of each other. Not always. Like for instance here, you have a body patch at this particular position, but you don't have a face patch. But if you go a little bit more posterior, you see nicely the face patch. In fact, this here is MSB here, and this is so-called MF, and this is ML. And also these face patches, they have some names. And this is our MSB I was talking about, which is very close to one face patch and here another face patch. And also here anterior, this here is ASB, this is AL face patch, and this here is another face patch called AF. And then very anterior, we have also another body patch. I was not talking about that except at the beginning. I told you a little bit that there was also a third one. This is this third one here, which is just lateral to a face patch called AM. But you see that they're very close to each other. Now, in the real world, when we see a face, we usually see also a body. It's very rare that we see only a face without a body in the real world. So that basically, well, one can ask then the question, yeah, are faces and bodies, headless bodies, processed completely separately? Well, we know that they're like largely non-overlapping networks of body patches and face patches. We know that from microstimulation experiments that we did a couple of years ago, in which we put an electrode in one of the body patches and then drove a current there, so we didn't record, but we microstimulated them, and then we see activation of the more anterior body patch in a scanner. This is an fMRI electrical microstimulation. When we stimulate the face patches, we see all the face patches activated, but we don't see the body patches or little of the body patches. So there's a partially independent analysis. However, we know from some recent work, like from Livingstone's lab, that if you show here a person in which the face is masked, the face is occluded, when the monkey is looking here at this portion of that image, face cells respond very strongly. So you can have responses in the face patch, in this face patch ML, in fact, to occluded faces. Also, we know from fMRI studies from Vogels that fMRI activation to monkeys, by monkeys I mean a monkey and its head, a monkey, well, the monkey body and its head, so face and body combined, that's stronger than the sum of activations to the isolated face and isolated headless body in a particular face patch here, in this face patch, in the Vogels of the STS. So that's why we asked, okay, well, are there particular regions that are activated more strongly to full monkeys, bodies and faces together, than to the face and body part? So we have here a new stimulus set in which we have monkeys, headless monkeys, and then isolated faces, so only faces, and we have a set of control objects matched in low-level properties. And then when we then look at the monkeys, and these are data of one animal, and we, so basically we subtract activation of the object from that to the monkey, then we see nice activation. And this could be a body and a face patches. And in fact, what we can do then is we can superimpose the face and the body patches and on those activations, and there is a little bit green remaining, but not much. So indicating that there is maybe here in the upper bank of the superior temporal sulcus, you might have a region here which responds more strongly to monkeys compared to only faces and only bodies. And in fact, to visualize that, we subtracted the bodies from the monkeys and the faces from the monkeys, and then we superimposed again the faces and the body patches. And you see here, indeed, here in the upper bank STS, these are data from 2 different monkeys, you see a patch, a small patch that is activated strongly for images of monkeys and seem to be less activated, at least not significantly activated, for bodies, isolated bodies without a head, and for faces. So it could be that there is a region there selective for whole bodies, but we have to do more analysis to substantiate this. This is really ongoing work. Now, and before closing, I would share you another experiment, which is basically addressing a similar question. Is we can have a monkey like here, in which the monkey, well, head is where it's supposed to be, but we also can displace its head to an unnatural configuration. It's always touching the body, and that we can compare them with only the body or only the face. So we have a natural versus unnatural configuration, and when we subtract basically natural minus unnatural configuration, we again see regions activated, so in which there is more activation to natural compared to unnatural, and they overlap remarkably the face patches. So it's as if we have, in some face patches, we have a stronger modulation, a stronger response to a monkey when its head is at a natural configuration compared to an unnatural configuration, while it doesn't overlap at all with the bodies. But again, this is something, an interesting result that we have to analyze in more detail. And then I want to conclude with something also I started with, in fact, is I at the beginning I said, when I was talking about the relevance of bodies for recognition, for actual recognition, for instance, that that we can derive a lot of information, especially from dynamic bodies when they move. So all the work that I have presented thus far, and basically most of the work also even in human imaging on body representations is done using static images of bodies. But of course we can also have dynamic bodies, as you can see here. So we have a whole set here in which we have we have faces, we have here bodies that move, we also have control objects that are moving, and then we have some scrambled controls. And basically, when we show these dynamic bodies and these moving bodies, and these bodies, the monkeys that act and they have a blurred face, then we see strong activations, and we see activations that we didn't see with static bodies, especially in upper bank. We see upper bank regions, like you can, like here for instance, here and there. So in the fundus and also more at the lip of the upper bank, basically, you see here you can see it also. You see well, patches that we didn't see for the static bodies. So we see more, especially in the upper bank of the STS. We see more body patches when we start using these dynamic bodies. And that's something we also want to analyze in more detail. In fact, what's also interesting, if we go to the MSB here, which we also see activated for the static bodies. So if we look at here this mid-STS body patch, the MSB, that's also activated when we use these scrambled bodies. Although it's very difficult to see a body there, but it contains some low-level features, and these MSB neurons, to some extent, are still responsive to those. But it needs to be verified also in some other animals. These are just data of one animal. As we speak, we are scanning a second one, and but there are very exciting new results, and the plan is basically to look at these more into these dynamic images, because we see more body patches, especially in the upper bank of the STS. We want to see which motion and shape features are present in these different patches, the interaction between the patches we want to study, and also, of course, we're also very interested in the causal contribution of these patches to behavior, which is still an open question. So this is where I would like to conclude, and I thank you for your attention, and I'm happy to take questions. And these are my collaborators here, some of who did the actual work. Okay. Yeah.
Okay. Thank you very much, Dr. Vogels, for the nice talk.
Yeah. So there is one question about from Bahare Tagisade, and the question is: are these body selective areas connected anatomically or functionally to areas related to movement planning, control of areas with proprioceptive inputs, or their activity is only associated with perception of feature of objects or body postures? That's a very interesting question. In fact, frankly, we do not know. Well, in the monkey at least, we do not know. But we know from other work that areas of the superior temporal sulcus, that they are related to or project to like parietal cortex, also prefrontal cortex, or premotor cortex, basically. So areas that are part of that so-called mirror neuron system. So these STS areas, they are part of the mirror neuron system in the sense that they give the visible information to the mirror neuron system. But it's the case that these cells themselves, at least in monkey, are not, they don't have the mirror neuron properties, so they don't respond when the monkey is doing a motor action, and when, as far as we know at least. But they respond to visual images of bodies, and that information is then sent to the mirror neuron system. That we know from work that has been done here, in fact, in our group by Koen Nelissen and colleagues. I don't know whether this answers your question, but it's a very interesting question. I should say, when we were stimulating, do microstimulation of the body patch of the posterior body patch MSB, the middle STS one, all the activations we saw were within the visual system. So we didn't see anything outside the visual system. But that doesn't mean that there are no projections outside the visual system, because if you do it for face patches, you get a very similar result, and we know there are projections from outside the visual system.
Yeah, maybe I wanted to also ask a question, if possible. So, this is about the relationship between the body patches and face patches.
Yes.
I'm wondering. So you've done this kind of a bubble approach to find the exact features of the body that the cells in MSB respond to. I'm wondering, have people, because it looks like there's some fundamental difference between face patches and body patches in the sense that if you really disturb too much of the face, face patches might not respond to it. And I don't think people really have shown that, like if you just show the nose, for example, to the face patch, does the face patch get activated? But basically, my question is, have people looked at this type of a bubble approach for face patches to see if face patches have been selected to particular features rather than faces? Yes,
that's a good question. And in fact, two groups have done that. One is published, the other is not published. I don't know whether it ever will be published. So there is a published work by Isa and De Carlo from Jim De Carlo's lab at MIT. They used the bubble approach to look at the shape, the face features that cells in a very posterior face patch responded to, namely PL. So you have a very posterior face patch, which is a little bit posterior to MSB, in fact. And they found that especially when you look at the initial part of the response, that face patch was very responsive to fragments, small fragments of the face, namely the eye. So it preferred the eye. But the eye had to be present within a contour. Well, there should be an outer contour there, like the boundary of the face. So if you just present an eye in isolation, the response is not that good. So the best response is basically an eye combined with some outer contour. So like there is perhaps some configuration information there already. And then there is work by MSB. They also showed the recording. Yeah. So Doris Sauer, she that's the second group. She used bubbles in MSB, and she had a poster at SFN, but I must say I don't recall really exactly she was not very happy with it, that I know, but you should ask her what she found. I know that Jim in Isa and De Carlo paper, they also showed some recordings in MSB, and also had some eye-related responses there. But it's interesting to an interesting technique. Now, bubbles is also now, it also has its restrictions, or limitations, of course. But yeah, for face patches, but it could be that configuration information might be more important for faces than for body cells. Although I have to be careful here, because in the bubbles that we used, there was some configuration information present, because we added several spatial frequency bands together, and you always have some reference of some body-like shape there. Parts of it are missing, of course, but it's not that you just have a single isolated part.
Okay, thank you so much.
I'm not making the claim that there is zero configuration information in a body patch. I cannot make that claim.
I see. Okay, thank you very much for the response. Is there any other questions?
Yes.
Okay, great. Okay, thank you for the wonderful talk. So I was very interested in the dynamic bodies. So actually for faces, that's also a very interesting question. And as far as I know, at least in the human, dynamic faces produce larger activations than static faces. And there is a difference in the distribution. So posterior STS seems to be largely involved or preferentially involved in dynamic faces. So I suppose I was and the STS gets its input from MT. So I sort of have two sort of general points. The first is it would be very interesting to know, what the distribution is for these dynamic faces, dynamic bodies that you showed, particularly the responses to the MSB neurons, which I think you showed were selective for posture. So posture, I suppose, is a static version of a sort of dynamic social interaction. And the second question was whether you used any sort of dynamic connectivity or interaction to see how the dorsal stream inputs from MT and other areas may affect these dynamic responses.
Well, I can answer immediately to your second question. No, we haven't done that. This is also block design. Huh? So, but yeah, we could do that. For your first question, now, we plan to use, in fact, we are going to do that. We are going to use in collaboration with Bea de Gelder from Maastricht. We are going to use the same stimuli, the monkey images. We also have human versions. We also have dynamic human bodies and human faces. We also have those, but we still need to scan them. But we are going to use monkey bodies and human bodies and human faces, monkey faces, both in humans and monkeys. So we can compare for exactly the same stimuli. We can compare the activations in both species.
Okay,
so that we are
very interesting.
Now, with respect to the extra activations that we see, like in the upper bank of the STS, which might be related to let's say posterior STS in humans, is the following. That's basically a general comment I would like to make, and it confuses some people, especially in the human imaging world. Is that as it seems to be the case based on all the monkey fMRI data we have, and when we compare it to human fMRI data. And now I'm talking about monkey fMRI data, not only about bodies, but also about faces, about scenes, color, and so on. That the upper bank of the monkey STS appears to be the homologue of the STS in humans, while the lower bank of the STS, where the face patches are, or most of the face patches are in the monkey, and also most of the body patches are in the monkey, that appear to be the homologue of more ventral lateral regions in the human brain, but not the STS in the human brain, right? So that might explain that the upper bank STS, which also in the monkey, which gets strong input from motion areas like MT, MST, FST, that also responds very strongly to these dynamic bodies and also to dynamic faces, as we know from other studies. So that might be the homologue of the human STS areas, while like the face patches that are and body patches that are well activated by static bodies and also by dynamic, but also by static. You don't need dynamic bodies to get strong activation there, like MSB, ASB, and the classical ML and so on, AL and AM face patches. Well, they probably are the homologue of like OFA and fusiform face area, so the more lateral one. Although part of them are in the monkey STS, but again, monkey STS, lower bank of monkey STS does not correspond to the human STS. So that's very important. So areas in human STS are probably homologue to upper bank STS in the monkey. So there's an anatomical distinction there. But I don't know how well I answered your question now on that.
Okay, Ian, if you're done with the question, we can have a very quick question from Ali Atai, and then we have to go basically close this session because we're a little bit over time. So Ali, if you want to ask your question briefly.
Hello, thanks, yes. Thanks. Hello, Dr. Vogels, and thanks for your nice lecture. I just come with an idea. What about using a back view of a complete full body with head, or a side view of a complete and full body but with rotated head instead of the bodies with corrupted head?
So you mean a back view of the head, of the body, you mean?
Yes, yes.
But
in order to make it seem natural.
Yes, it looks so. And what's the purpose? So you have a natural, configuration, but you just show it from the back. Right?
Yes, yes. In order to not to have the problem with the face, not to activate.
Yeah. Okay. Now, in fact, like for the dynamic bodies, what we have there is we blur the face. So I don't know whether you noticed on the videos, but the face is blurred. So and even for these blurred faces, we see activations in face patches. So I think that also from the back view, you'll have an activation in the face patch.
Oh yes. Okay.
Yeah, I think it's not sufficient to have zero activation in the face patch, even from a back view.
True. Thanks.
Yeah.